So the purpose of this video is to demonstrate a really simple experiment to test the functionality of one of these electrochemical H cells. And so we have both of these H cells sold by Lab Basics, both in the 50 and the 10 milliliter size. And there's a membrane that separates two halves, which are of particular use if you want to test redox flow batteries, like non-aqueous redox flow batteries, where you can have two halves of a cell separated by a membrane. And so this video is to show you a simple symmetric aqueous cell that you can use to ensure the functionality of the cell. So just some notes on membranes. So if you're doing a simple aqueous cell, we found that just using a Wattman glass microfiber filter, the 934-AH, works, but there's definitely crossover issues. So you need to do your cycling, your charge-discharge cycles pretty fast. For non-aqueous cells, you can use CellGuard, which is a common membrane material in lithium-ion batteries. You cannot do this in aqueous cells because they're too hydrophobic. We have a ton of this stuff in the lab that works for the non-aqueous, but there's still some crossover issues, so it's not perfect, but fairly low uncompensated resistance. The best choice is one of these anion exchange membranes. There's a variety of types, but you can buy them from Lab Basics. We purchased these squares along with punches that are of the same diameter as our cell, so we can punch a circular disc that would then fit in between the two halves of the H cell. So just a little context, the idea is if you wanted to build, say, a non-aqueous redox flow battery, you can look at the individual cyclic voltammetry data for each of the individual redox transitions for the different compounds. And so what I'm showing you here are data that are overlaid for an iron 3+ acetylacetonate compound in a non-aqueous cell, acetonitrile, with a 0.1 molar TBAPF6 background electrolyte. And so that's the red line here, and you can see a really pronounced transition from the 3+ to 2+, quite reversible. And then we have a sort of matched compound, but the chromium 3+ version in blue, and you can see more transitions. So the idea is you could build a battery out of really any of these different transitions, where in one half of the H cell you'd have the iron compound in a particular redox state, and then the other half you would have a chromium compound in a particular redox state. You could also build a battery out of chromium in one redox state and then chromium in the other redox state in the same H cell. And so I'm showing you here a bunch of different options, just looking at the voltammetry here and the different transitions that you might have. And this is captured in this PowerPoint that's called H cell notes. So as one example of the battery, we could say use an iron chromium battery in the 1.6 volt theoretical cell voltage. And so that would be using this iron 3 transition for the iron compound, and then this chromium 3 2+ transition over here. So this is a logical starting point because this is kind of reversible, this is definitely reversible, and there's like a reasonable cell voltage here. And so I've got a really general energy diagram here. The main challenge in this particular cell practically is that the iron 3 ACAC and the chromium 3 ACAC both start in the 3+ oxidation state. So in terms of the solid compound, they're both in 3+, and so we need one of them to either be in the 2+ and the other to be in 3+, which means you have to do a pre-electrolysis step. So you can convert the iron 3 into 2+ by reducing it coulometrically, or you could do the same for the chromium. But effectively what you have is the charge state. We have the chromium in the 2+ state, so it's highlighted here. So there's an electron in this orbital, and because this is a higher energy compound, it's out here at more negative potentials, then this would galvanically descend. This is the natural discharge state, where the iron 3+ in the other half of the cell would receive that electron. So that's the charge cycle. That's the discharge cycle in the charged initial state, and then we would charge this back up in the opposite direction, the idea being that there's roughly 1.6 volts between these two states. And this slide captures some general notes on how you might pre-electrolyze the original iron 3 ACAC into an iron 2 state using the same H cell. So we did this with essentially a 3-electrode cell. This is there's a more updated version of this, but we effectively took the iron compound, the iron 3 ACAC, in the left side of the cell with the working electrode. We used carbon rod here, and we actually put the non-aqueous reference electrode also in this same side. And then on the other side over here, we filled this with a ferrocene solution. I think we used 20 millimolar, same electrolyte concentration and composition in both halves of the cell. and then we electrolyzed this. And so this was a big counter electrode. We used a big chunk of platinum on this side, and we just drove the reaction here, and we just used the ferrocene as an electron donor, effectively a sacrificial redox. And we pulled electrons through the circuit over here to make the iron 2 version of this. And so then if you wanted to make the actual original battery we were talking about, you would leave this new iron 2 plus ACAC in solution in this side of the cell, and then exchange this side with the original chromium 3 ACAC solution, again matched electrolytes. So this is like the non-aqueous version of this cell, but the impetus of the subsequent part of this video is just to show you the aqueous symmetric iron chloride cell that we used to just make sure that the cell was working as we thought, all the stirring was working as we thought, and that the chronopotentiometry, the sort of charge-discharge measurement, was all working accordingly. Of course, the solutions are different and the parameters are different, but this is a good preliminary check just to make sure everything's functional. So this is a lot of background to really show you just a much simpler cell. So the actual conditions for the iron-iron aqueous test cell come from this article here. It's not exactly the same because they're actually doing potentiostatic cycling rather than galvanostatic cycling. This is a figure from their data, which is essentially the same thing that we would be expecting to capture in our chronopotentiometric or CP trace. So this is the starting point that we used for building the iron-iron aqueous symmetric cell that we used for testing. And then here are the actual conditions that we use, which are going to be restated in the subsequent video, which shows you actually how to do this practically. But this is the reference conditions all in one place.
Before preparing your H-cell, find the maximum immersion surface area of the graphite electrode. To find the surface area, we measured the diameter of the graphite rods using a micrometer and calculated the radius. Although our reference data used a surface area of 4 cm squared, our specific cell size allowed us to use 3 cm of the rod, resulting in a surface area of 6 cm squared. Then, we made a stock solution of sulfuric acid, iron 3 chloride, and iron 2 chloride. We used a tetrahydrate form of iron 2 chloride, so we accounted for this in our calculations for this solution. We used 1 molar of sulfuric acid, 0.05 molar of iron 3 chloride, and 0.05 molar of iron 2 chloride. Take into account the original molarity of your sulfuric acid for your calculations. Next, since we are using the smaller H cell, we will need 11 milliliters of our solution for each arm of the cell. Here we are pipetting 10 milliliters in one graduated cylinder and 1 milliliter in another graduated cylinder before adding both to a single beaker. We then repeated this process into another beaker for the second arm of the H cell. We began H cell assembly by first making sure our graphite electrodes were thoroughly cleaned using DI water. Sandwich a Wattman glass filter between 2 silicone rings on the H cell. Push the cells together as tightly as possible, but carefully so that the silicone rings and the filter do not drop out. Next, cut a small piece of parafilm down the middle and wrap it around the middle of the H cell to help prevent small leaks during your experiment. Attach the clamp to secure the cell and straighten the H cell as much as you can so it stands flat and not tilted. Next, add your stir bars. Add in your iron solution simultaneously. Next, securely cap both sides of your H cell. Turn on only the stirrer of your stirring hot plate. Make sure each side of the cell is stirring, bubbling, and the solution is at equal heights on both sides of the cell. After 5 to 10 minutes of degassing, carefully take both pipettes out of the cell. As you are degassing, you can add the caps to the top of your graphite electrodes near the indentations and rinse with acetonitrile. Twist both caps onto each side of the H cell. Double check that exactly 3 centimeters of the graphite electrode is in solution. Connect the working electrode clip to one graphite electrode and the reference and counter clips to the other electrode. To run your experiment, open the CH instrument program and run an IR compensation test to determine the resistance to account for in your chronopotentiometry test parameters. We like to run the IR test 3 times to check that our resistance is consistent. This is the value we are looking at. If this value is very high, you may want to adjust your cathodic and anodic current values. For reference, our resistance was around 19 ohms. However, for this demonstration, we are not actually running an iron-iron H cell, so these values are extremely high. Now, run an OCP for around 1200 seconds. Once your OCP is done, make sure you save your data into its own folder and that it is clearly labeled so you can know what to expect to see before ever opening the file. For our experiments, we like to start with the number of the trial, what tests we are running, and what our conditions were. Now again, we are not running an actual H-cell test here, so for reference, here's what our OCP data look like. Following our reference data, we used a 2 mA current in our IR loss calculation. We multiplied our current by our experimental resistance to get around a 0.04 voltage drop, which was well below our 0.10 to 0.15 voltage limit. The anodic time used in our experiment was calculated using Faraday's law based on a single electron redox event, a 0.1 molar concentration, and an 11 milliliter volume, resulting in a full theoretical capacity of 106.1 coulombs. To shorten the screening run and simply verify that the experiment works, we can run our experiment at only 2% capacity. This reduces the target charge to 2.12 coulombs, and after dividing this by our 2 milliamp current, we get an anodic time of 1060 seconds. After running OCP, you will run your actual chronopotentiometry test. For the iron-iron aqueous test cell, we used a cathodic and anodic current of 0.002 amps, a high E limit of positive 0.3 volts, a low E limit of negative 0.3 volts, a cathodic and anodic time of 1060 seconds, an anodic initial polarity, and a 3-second data storage interval. We ran 4 segments and used time as a current switching priority while keeping auxiliary recording off or unchecked. Again, because our resistance was so low, we kept these original parameters. Had our resistance been higher than 75 ohms, we would not have been able to use our 2 milliamp current and would have had to divide 0.15 volts by our experimental resistance to get our max current. Again, we are not running an actual H-cell test here, so this graph is not accurate. However, for reference, here is our data. Finally, remember to always save your data after your experiment.